Reimagining resolution at single cells with generative encoding. What can you do with generative encoding? Imagine if you took a single cell and its corresponding gene expression vector, copied it 3 times, and changed its gene expression or set of genes for each of those copies in different ways. Because you can transform those gene expression perturbations into tiles, this is essentially what we've done here, and we can see that those 3 copies from the left to the right change in a certain way. So that is what generative encoding does. It enables an alignment and then a transformation between modalities or datasets. So let's go into a bit more detail. It's inspired by deep learning, which learns from large samples with many parameters. It's the encoding of data into a reduced dimensional space. So if we take the expression and the datasets y and x, or in some situations y equals x, we can learn a set of parameters k, v, j, and u that allows an alignment between y and x into a reduced dimensional space of d1 by d2. Let's look at one part of the equation. So if we look at the parameters and the data of y or x, we can see that the sample parameter and the feature parameter on either side of the data set wraps around and transforms the data into that subdimensional space. So the good question is, how does it work? Well, it's very similar to a method called Procrustes, which is a geometrical method that aligns images via rotations. If we look at the image on the left, we can see a red heart shape and a blue heart shape. So when we use Procrustes, which is on the right, we notice that the images match and the shapes align via a set of rotations. Similarly, generative encoding does something in the same manner. The parameters that generative encoding learns are rotations, and we'll show later that it can be transferred or combined. Now, one important matter is that the solution of the generative encoder is not identifiable in the statistical sense, given that there are many possible rotating solutions. For example, if we rotate the current generative encoding plot 9 degrees to the right or clockwise, we'll notice that while the points match up, the solution is not the same, and thus explains why it's not essentially identifiable. An important part of the generative encoder is the transfer learning part. Essentially, the aim is to reuse a pre-trained model and transfer the information or the parameters which aligns the 2 data sets and transfer that alignment onto a third data set. For example, if our 2 data sets are y and x, and we learn the parameters k, v, j, u, and we have a third data set z, how do we transfer the parameters? Well, if y is very similar to z in the sense that the axes are identical in some way, for example, if the samples were the same size or the feature set was the same size, then we can transfer the corresponding parameters. If the feature set was the same between y and z, we can transfer v. If the sample size was the same, we can transfer k. And by doing so, we transfer the alignment that is learned in the first pre-trained model that encodes the information between y and x, and transfer that information to z. After we transfer those parameters, we then learn a new set of parameters that are highlighted in orange. That can be L or W. Now, how do we know that this can work? We've tried this on a spatial transitomic data set, and the representation of the histology slide is on the top, is on the bottom left. We notice that there are three types of cells, WT1 marking podocytes for pink, CD45 marking immune cells for blue, and PNCK marking renal tubules for yellow. Now, what are the 6 tiles in the middle? Well, these are the top 6 components of the parameters that we learn. So if we take K or V, in this case, V is the feature parameter. So we'll take V, which is of dimension pixel by reduced dimension D. And if we take the 6 components of V and then convert it to images by rearranging the vectors into matrix format and then plot those matrices, those are the top 6 components that we get out from the reduced dimensions. Now notice that these images, the top 6 components, are very similar and correlate to what we actually see in the histology slide: pink, yellow, and blue. So now that we know that we can transfer parameters, how do we combine them? Can they be combined? Well, if you learn the pre-trained model that relates y and x, and then have a third dataset z, you can learn the mapping between y and z by passing on the parameters by transfer learning and learn a new parameter that's called w. So in this case, u and v correspond to y and x respectively, and w corresponds to z. So once you learn those 3 set of rotation parameters, you can then combine them using linear operations such as a dot product, and then combine those rotations to get a super rotation that allows you to transform those data sets into the same dimensional space. For example, if we take the bottom plots and the first plot, which is of 1 rotation, we notice that the plot does not encode the information that allows you to structurally see the cell types, which is the main component of the data set. When you increase the 2 rotations, the structural shape of the data begins to appear. And when you go to 3 rotations, it is quite evident. So in this case, 1 rotation corresponds to U, which is just the first rotation. 2 rotations corresponds to U and V, and 3 rotations corresponds to U, V, and W. Now, the combination of these rotations allows for a richer, more structurally aware visualization and transformation of the data. And so we can sort of see that adding in more rotations allows for a better transformation. And so if we compare this with deep learning that has the composition of functions, we can sort of relate how the combination of rotations and sort of perhaps educationally guess how deep learning could behave through the analogous comparison between rotation combining and function composition. So that was something I found interesting, and I thought I would share that. One other interesting concept was the fact that when using SVD or PCA initialization, the components, when taken in rank order, increased in variation. So the top row corresponds to PCA initialization, and the bottom row corresponds to SVD initialization. This is of spatial transcriptomic data, and the components represent images or cells. The first plot on the left shows the 5th rotation, And we notice that it's a diffuse, large globular green shape, which corresponds to, for example, a cell. Now, when you move to the 25th rotation, you notice that the number of cells increases, and so does the variability of that matrix, which is actually a vector in the component space. So that looks like about 5 cells. And when you move to the 45th rotation, it's about 10 cells. And moving to the 65th rotation, it's about 15 cells. So the variation definitely increases as you increase the number of rotations. And this is consistent as you increase the rank and visualize each component in that ranking order. So now that I've described generative encoding, how it works and what it can do and how you can use it, we'll go to the main core of the talk, which is using it on spatial transcriptomic dataset. So let's say, for example, you had a kidney single-cell RNA-seq dataset, and you could align it to the spatial transcriptomic information, that is gene by spot or tile. Aligning this tile of transcriptome information to the actual image, and then aligning that image to unseen histology image allows you to create a mapping or alignment between kidney single-cell RNA-seq to unseen histology. And that's what we see on the right diagram. So this is what happens when you align that full kidney single-cell RNA-seq to the spatial transcriptomic data set and then onto the histology. If we look at the main plot, we can see that it's of a immunofluorescent spatial transcriptomic image. When we separate those cell types by color, we notice that the immune markers are at the top, WT1 is in the middle, and PanCK is in the middle and bottom. Splitting this main image up into 64 by 64 parts or tiles allows us to convert each tile to an alignment that aligns between that individual tile and the gene expression. The gene expression of the single-cell RNA-seq is then mapped to those tiles, and we can then visualize them using a gene expression heat map. For example, we take HLA, which is an immune marker, and we notice that it covers most of the immune cells on that histology image. So that's the main part of the alignment. What happens if we can inverse the transform, inverse the alignment, and be able to visualize single-cell RNA-seq as an image? That's what we see in the top right-hand corner in the first set of plots that I described at the very start of the session. When moving from the left to the right, as I described earlier, we are increasing or perturbing a set of genes and visualizing that single cell as a tile, which I find quite interesting and revealing. So we know that we can convert a single cell RNA-seq cell to a spatial transcriptomic tile. What happens if we could do this on a scale that allows us to explain cell type interactions in space? We know that gene ontology is for biological function of gene sets and that cell ontology is to define and characterize cell types. If we could use this information in some form and be able to explain cell type interactions in space for spatial ontology, that could allow biologists to interpret and use the current information and databases out there to be able to make and generate hypotheses that allows us to gain deeper insight into the biology. Now, with those QR codes and the YouTube links, you can start to get a sense of what is happening with this generative encoder and can start to see how the generative encoder could assist with some idea or concept of a spatial ontology. That wraps it up, and I'd like to acknowledge the following people for database dashboard work and discussions, advice, and talks about strategies and techniques. And also importantly, I'd like to thank these people for inspiring the work, for building up a certain way of thinking and allowing a certain direction that has helped me get to where I am, or where this model is today. Thank you.